So much, Murray. It's totally an honor to be a part of this really cool event. So I'm really happy that this is getting going. So I think we really need the front lights down the most. Perfect. Thank you so much. So I was getting ready to give this talk, and I was thinking to myself, okay, what lights my fire? What do I love more than anything else in the world? And I had to be honest with myself. I came to the conclusion that I love brains. I love brains kind of a lot. Now, before you get worried that there's a zombie at Northeastern, this is not the case. I don't love to eat brains. What I love to do is study brains. And so what you're looking at is a picture of me from about 5 years ago, and I was super excited because a photograph that I had taken on a microscope of a neuron was put on the cover of the journal called Brain Research. And that made me really happy. It's always exciting to be on the cover. And what I did between then and now is I took that technique that allowed me to take that picture and I brought it here to Northeastern, and now I can show you a picture like this. So this is a 3D image of a single neuron that one of my graduate students took. And the way that she created this image was she punctured a neuron, a single cell, with a very, very tiny glass needle, turned on a current, and filled that neuron with a fluorescent dye. She then imaged it using this really beautiful, fancy microscope that we have, and that allows us to create this beautiful image. And I have been looking at images like this now for about 10 years. I still think it's one of the most beautiful things I've ever seen. And one of the person I probably really have the most to thank for this image existing is someone I've never met. It is Santiago Ramon Cajal. He is widely considered to be the grandfather of neuroscience. And the reason for that is that he spent his career, he was a Spanish neuroanatomist, and he spent hours and hours and hours at the microscope looking at tissue from all different kinds of regions of the brain, and then by hand painstakingly redrawing what he saw under the microscope. And his images are now iconic, and they are totally beautiful. So I'm going to show you a couple of those images. So first, this is an image that he drew, again, by hand, just by looking in the microscope, right? There's no photography back then. This is sort of mid to late 19th century, or at least there wasn't microscopic photography. So here he was able to show people for the first time really what some of the layers of the cortex look like. He also was able to draw the hippocampus and its sort of signature trisynaptic circuit that you can see there. He also looked at the retina. Retina has really distinctive looking cells and distinctive looking connectivity, and again, I just think they're so beautiful. And finally, one of my favorites is this positively haunting image of Purkinje neurons in the cerebellum. And what you can see is that in each of these images, the neurons all look really distinct from each other, and we can see for the first time how differentiated the brain is in different parts of the brain. What Cajal was really famous for, though, what he really gained the notoriety for, was that he was the first person to describe neurons as being distinct cells. Just like every other cell in the body, neurons exist individually. They are not physically connected to each other. The synapse ideas came later, but Cajal was really known for being the first person to describe neurons as individual units. Before that, all of the theories thought about the nervous system as a single continuous web. It was called the reticular theory. In contrast, Cajal's theory was called the neuron doctrine, and he ultimately won the Nobel Prize in 1906 for this discovery and his description. Now, he did not get the Nobel Prize all to himself. He had to share it with this man Camillo Golgi. Golgi was famous for a number of different things. As you might imagine, he is the namesake of the Golgi Apparatus, part of the cellular machinery. And Golgi was actually one of the real proponents of this reticular theory, which, as I just mentioned, was wrong. So why did he get the Nobel Prize too? That doesn't really seem fair, does it? So the reason he was part of this Nobel Prize was that he developed the technique that allowed Cajal to see what he saw and to then ultimately describe it. So what Golgi figured out was that if you treated brain tissue with a silver solution, what you would be able to see was some, but not all, of the neurons. The silver solution, which then became known as Golgi technique, would label a subset completely randomly of the neurons in the brain. And what you can see is that, so here's that hippocampus again that I showed you one of Cajal's drawings of. If we zoom in, you can start to see that you can really see individual neurons. So what's amazing about this technique is that it labels neurons in their entirety. It just doesn't label all of them. And it allows you to see very nicely the overall structure of neurons as well as general architecture of the brain. If you zoom in even closer, you can even see dendritic spines, which we now know are the receptive areas of neurons where they receive information from other neurons. Now, as I mentioned before, this technique randomly labels a subset of neurons. This technique is still used very much today in lots and lots of neuroscience labs. But what's amazing is that this technique is over 100 years old and nobody knows how it works. Even today, we have no idea why the neurons that get labeled, and the ones that don't get labeled, are excluded. And so that it becomes a limitation in terms of understanding what kinds of neurons you're actually looking at. However, in the last couple of decades, there have been some major advances on that front, and they have also helped us generate some really beautiful images. And so we have made advances with the help of jellyfish. So jellyfish, as you probably know, glow in the dark, and they do that because they contain fluorescent proteins. And what scientists have been able to do, people who also won the Nobel Prize, by the way, is harness the genes for those proteins and find ways to express them in all different kinds of species and cells, and this allows us to see reds and greens and blue in different neurons. Now, what this allows us to do then is now we get to choose. We get to choose exactly which neurons we want to look at. In my lab, the neuron that I showed you, the spinning neuron, was labeled specifically because we know it projects from one area of the brain, the prefrontal cortex, to another area of the brain, the amygdala. Now we have a lot more control and power over the kinds of neurons that we're investigating, and we can try and learn about how their structure relates to other things. So just to give you a couple of other examples of how these fluorescent proteins have changed neuroscience. So here are those Purkinje neurons again, which this image is totally beautiful in its own right, but we just are looking at the neurons. In contrast, now with fluorescence imaging, now we can look at Purkinje neurons and look at them like this. And what you can see is not only the neurons themselves, which are there labeled in green, but also the structures and the cells that surround it. Things like scaffolding proteins, glia, astrocytes, which are what's labeled in red or magenta, as well as the granule cell layer, which is there in blue. And so now all of a sudden we can see neurons in context thanks to these kinds of techniques. As another example, here's that Golgi hippocampus again. It's nice we can see what it looks like. However, using fluorescence, we can now look at the hippocampus like this. This is an image of the hippocampus from a mouse that has been genetically engineered to express fluorescent proteins in all of its neurons in a random pattern, so that we can now look at all different kinds of neurons. We can see how they project, where they project to. You can imagine that if we just labeled all of the neurons the same color, we wouldn't be able to get a whole lot of information from that. Now that we're able to label neurons with many different colors, we can separate the pathways and try and understand better how the brain, or how different areas of the brain connect to each other. This mouse is called Brainbow, and was, yes, how could you not name it that? This was developed a few years ago and made a big splash. As you can see, I don't even think it needs any explanation for why. It's just totally stunning. And so what is amazing to me is that for my job, like I get paid to do these things, as a member of the psychology department, I can use some of these techniques to try and understand the neurobiological basis of behavior. So what you're looking at here are two rats. Oh, there they go. Okay. That are exhibiting very different behaviors to the same stimulus. So both of these rats have been conditioned that when they hear a specific tone, it means that they think they're going to get a shock. And so what we look at is differences in fear behavior when these animals hear the tone. What you can see is that the animal on the left is freezing, right? It's hardly moving at all. It may be, so these are on loops. But when the animal hears the tone, it startles a little bit and then it just freezes. That's a really common response to a fear-conditioned tone. In contrast, the animal on the right kind of looks like he wants to escape, right? So this animal is not having this passive freezing response to the fear-conditioned tone, but is having an active response. And what we're really interested in the lab is, are there neuroanatomical differences in rats that freeze versus rats that try to escape? And so what we can do is take the brains from these animals and look at their neurons. We can look at their neurons in the brain regions that we think are involved in these kinds of fear processes and say, are there any neuroanatomical markers in neurons that are part of specific circuits that are driving these behaviors? What we're also interested in lab is sex differences in these kinds of responses. So are there neuroanatomicals that are distinct in male versus female rats that indicate these kinds of behavioral differences? And so this is the kind of thing that is applicable to human studies, to things like PTSD, and the kinds of work that we are doing to try and understand what these markers are relevant to things like DTI or diffusion tensor imaging in humans, which can look at connectivity between different brain regions and look at patient populations versus healthy populations and see whether the kinds of differences that we see in the lab are also translatable to humans as well. So that's basically all I have, but I sort of thought of a little analogy. So I think there are a lot of people who maybe say, oh, I want to be an astronomer because I just love to look at the stars all day. And then you go and take an astrophysics class and it's just a bunch of math. And you're like, wait, where are the stars? In contrast, if you love looking at neurons, that is really your job, if you're a neuroanatomist, is to just stare at these beautiful images all day. So that's why I love it. And that's it.
Time for one or two questions.
I guess maybe I should have this on for.
Yes.
You said that so you would be able to see differences in the two rats that had different behaviors. Do you know what kind of visual differences you'd be looking for?
Oh yeah, so the question is this still on?
Yes.
The question is, what differences would I be looking for in terms of trying to compare animals that have different kinds of behavior? And the answer is, what we look for are patterns in the morphology of the neurons themselves. So what we can do is look for differences in the way the neurons branch. So some neurons might have lots of different branches, which indicates that there's greater complexity. We can also actually look at the spines and the morphology. So there are certain types of dendritic spines that indicate that there's a lot of plasticity going on, so which are relevant for things like learning and memory, or more like learning. And then there are other kinds of spines that indicate that there are stable synapses there, which might be more relevant to long-term memory.
Okay, one last question, yeah.
So since you're looking at the difference in sexes, what's the most unique morphology difference you've seen so far? Or what was the most surprising thing? Like either it was they were the same and you expect them to be different, or you expect them to be the same and they were different?
So I will say that probably the most surprising thing I've found so far was something I found before I came here, which was we exposed animals to stress. And normally what you expect, or what had been seen in males before, was that the dendrites sort of retract. And animals that have undergone stress lose dendritic material. They lose gray matter. And so we thought, well, women are more susceptible to stress-related mental illnesses, so we thought we would see an even more pronounced retraction in the female animals. And what we actually saw was that their neurons grew in response to stress. And so that was definitely, it was like my hypothesis is completely backwards, but that's science. And so now what we're trying to do in the lab is understand why males and females have these different structural plasticity changes and what they actually mean on a functional level.